So good morning. My name is Adrian Senderowicz. I'm a medical oncologist with almost 20-year experience in clinical development from the small biotech and large pharma, and also have significant regulatory experience as a medical officer at the Food and Drug Administration. I have the privilege today to be the chairperson for the program committee, and I need to thank all the organizing committees. It was a team effort, particularly to Cheryl Nass and her team, who did an incredible job putting this together. As Michael mentioned, this is the workshop called Policy Issues in the Development and Adoption of Molecularly Targeted Cancer. And let me first thank you for attending this, I think, great workshop. I'm sure that you will appreciate and have fun as much as we will have. And let me first share why we are here, what I believe, why we are here, and then to try to discuss a brief overview of the workshop. So let's go back to, can you hear me well? Yeah. Let's go back to 2000. Let's be younger or young again. It's nice to be young again. And let me focus with lung cancer, but we could have focused in many other tumor types, because what it went through over the last 15 years is incredible, I would say, as from an oncologist point of view. But we could have taken, for example, melanoma, breast cancer, medullary carcinoma, and we could discuss a similar discussion. So lung cancer was a simple or simpler tumor type from an oncology point of view. Basically, around 15% of tumors were small cell lung cancer, 85% were non-small cell lung cancer. Certainly, the therapies for either tumor type was different. And in the non-small cell lung cancer, about half were adenocarcinomas, and the rest were squamous carcinomas. And the way we treated, at least when I was a fellow at the NCI, it was very, I would say, unsatisfactory from our point of view. And this is the TAX-320, and I could have taken other drugs. I'm not picking any particular drug. But this was the pivotal study for docetaxel, or Taxotere, that's the commercial drug, against best supportive care. And as you can see, the median survival was less than 6 months, and the response rate in the active control, I would say, and actually the experimental arm, was short of 6%. As you can imagine, despite that, this drug was approved and is the mainstay therapy for second-line lung cancer. But since then, obviously, there were a significant revolution in the genomics and understanding of the molecular pathogenesis of cancer. And many companies start developing small molecule tyrosine kinase inhibitors that are directed against some important oncogenes. And over the last few years, we start seeing these kind of responses that something was almost, when I was a fellow, unheard of. I mean, you can see patients who have a baseline CAT scan with a very significant lung cancer, advanced lung cancer, and within a few weeks, very early and very profound responses. And at the beginning, we didn't know why this was happening, but what it was evident, and I'm focusing on one drug, but we can focus on many other drugs, that these responses were very, first of all, prevalent. Almost 60% or more than 60% of those patients have a response of this nature, very profound and very durable, in at least for our way of thinking. The median response rate for these patients were 48 months, so more than a year. And if you refer back to the slide that I showed before, the survival was half a year. So certainly, and this is the waterfall plot that shows how much the patient shrink their tumors. And this is the study that compare this drug, but again, there are other drugs. It's called crizotinib. It's an ALK inhibitor versus chemotherapy, showing that the median progression-free survival was around almost a year, 11 months. So something that we are, in this particular population, and in few other populations, we are seeing a very significant differences for these patients. So, and based on this understanding of the molecular pathogenesis, it became clear that not only ALK, many other important oncogenes are, driving tumors, and we can target those oncogenes with small molecular tyrosine kinases. You have EGFR, you have BRAF, MET, etc., and ROS. And so basically this subgroup or segmentation of these patients is very important because based on this segmentation, you can treat different patients with different agents. And how do you identify these patients? And this is based on, and I think is the crutch of the presentation today, is based on in vitro drug products. And these are tests, these are the design tests that will identify patients with the FISH ALK translocation. And you can also observe in this particular patient an overexpression of the ALK protein and the mutations that explains why this tumor has this rearrangement. So it's a very significant responsibility because of a false positive test that is not well controlled and not well designed will lead to unnecessary treatments. And these drugs, although maybe more safe than the chemotherapy, have their own toxicities. However, false negative studies will prevent patients who will need these kind of therapies to receive this therapy. So it's a big responsibility for manufacturers, for physicians who need to order this, for pathologists to perform this, for the regulatory agencies to regulate this, to be sure that when you go to see a patient, you know that if a patient has this positive test, they have at least 60% chances of having a response. So this creates a lot of challenges. This is a new way of treating patients. And it creates a lot of challenges from different stakeholders. I think the opportunity to bring all stakeholders here is to challenge each other, discuss each other. And I believe that based on this discussion over the day and a half, today and tomorrow, of course, we will have I think an important outcome is how to treat better patients in the future. So this is the presentations, and this is the session one will be, challenges in developing clinical biomarkers. We want to discuss the single analyte that I share, but then we have multiplex, and we want to discuss why multiplex analysis is important for patients. Then I will be chairing the session 2A, obviously the most important session of all. And then we want to have a lunch break. Then we want to have the issue of reimbursement, a very significant issue that we need to discuss, and how to generate genetic evidence based on clinical trials and other mechanisms. Then day 2, we want to have how we implement in clinical practice and the business model for test performance. So thank you, and there are no questions. I think we move to the first speaker. I think Samir is going to chair.